Welcome, everyone, to this week's Journal Club. So this week we are very lucky because we have Alessandra della Torre here from the University of Rome, Sapienza. So we've worked with Ale for a long time on malaria vectors, but her and her team also work on other mosquitoes, dengue vectors in Italy. But today we're going to hear about some work in West Africa on malaria vectors, on a long-standing conundrum that people have been thinking about for many years now. So this very interesting paper recently was published, and Ali is going to tell us all about it now. Just some housekeeping. If you could ask questions in the Q&A box at the bottom, there will be opportunities to speak at the end of Ali's talk. But if things come to you during the course of it, if you just write them in the Q&A box, that's probably the best way to do it. And also to let folk know this is going to be recorded. So yeah, just if you're all comfortable with that. I think that's everything. Are we good, Shri, to get recording and start?
Yeah, the recording's on right now. So yeah, over to you, Alessandra.
Okay, thank you very much, Chris. Thank you for inviting me to present these recent results that we get. Can you see my screen?
Yes, it's not on presenter mode yet, but I can see your screen.
Yes, now it should be.
Perfect.
Okay, so the focus, as Chris mentioned, will be on this recent paper where we describe a novel cryptic taxon within Anopheles, gambiae, and substrito. But and this work was done in collaboration with many collaborators also out of Sapienza University. But I would like here to present the group who work on this subject, and Beniamino and Carlo, who are here and will contribute to the question and answer session. And I took an historical perspective in introducing these results, because I think it's interesting for people who are younger and maybe new to the subject to have a little bit of historical knowledge on how we end up with so many species and forms described within the main vector species, malaria vector species in sub-Saharan Africa. So I'll start from the very beginning, more than 60 years ago, when Anopheles gambiae was considered to be a single species with very different adaptation to different conditions and varying degree of association with humans and of capacity to transmit malaria. In the 60s, however, in the framework of works that were devoted to understand insecticide resistance in this main malaria vector, and it's interesting to highlight here that more than 60 years ago already there were studies focusing on insecticide resistance. George Davidson was crossing different populations. You have to think that it was a time where there was no molecular tools available. So to understand the genetics of insecticide resistance, he was crossing population of Anopheles gambiae from different geographic regions, and he realized that some of these crosses didn't provide a viable progeny, but the males of this progeny were sterile, as you can see here from the testicle of one progeny of one of these crosses, which are clearly sterile. And this led to identify or to hypothesize the existence of mating types that were called ABC, Merus, and Melas, which already at the time were shown to have different ecological adaptation at the larval stages. But the real breakthrough came a few years later with the seminal study of Professor Coluzzii here in Sapienza University in Rome, my mentor, who by analyzing the polyten chromosomes of females that were in the process of digesting the blood meal, identifies some paracentric inversion, you can see here in the bottom, that were fixed in different populations, which he actually used, particularly on the chromosome X. He actually used as a first taxonomic use tools to identify these different species, which open the possibility to study their behavior, their actual distribution, their different adaptation, and the different role as malaria vector. And about 20 years later, with the development of molecular biology and genetics, a very important tool became available. This was produced by the group of Frank Collins, and I'll take here just a second to speak about Frank, who died two days ago. And I think he was a great scientist and gave a very big contribution to this and many other fields in the malaria epidemiology and entomology field. And the Frank group produced this very basic now that we see it 30 years later, PCR tool to distinguish the different species of what was called at the stage the Anopheles gambiae complex. And still this approach is used all over Africa to identify these different species. And based on this, the distribution could be clarified. And we now know that Gambiae arabiensis are the species with the largest range, while the other species have more restricted ranges. And also thanks to the availability of an easy tool to identify the different species, many other information on their ecology became available. But going back to the cytogenetic work, what coluzzii saw studying the polyten chromosome complement of Anopheles gambiae mosquitoes was that in addition to fixed inversion that could be used as taxonomic tools, many populations were, particularly in Anopheles gambiae and suscripto, were characterized by polyten inversion polymorphisms, which, as you can see here, were mainly present on chromosome 2. And for some of these inversion polymorphism, a clear adaptive value was shown. For instance, the best known one are allowing these mosquitoes to live in more arid conditions. So you clearly see a trend in the inversion frequency increasing from humid to dry areas. And he also realized that these inversion were not, I don't think you can really appreciate this picture, but just to show you that different populations in West Africa characterized from different geographic origin were characterized by different arrangements of a few of these polymorphisms. And that also, you cannot see here in the picture, but also there was a clear deficit of heterozygotes in some regions, and leading to the hypothesis that there were some populations sympatric but not mating between each other. And this led coluzzii to name 5 chromosomal forms for Savannah, Mopti, Bissau, and Bamako, which were characterized by some inversion arrangements, which could coexist in some cases in the same geographic region, but without mating between each other. And of course, this basing all this incipient speciation theory on inversion provided also an element of why this happened, because inversion polymorphism protect some sequence of genes that can be adaptive in some marginal situations. And but in the same time had some limitation because some of the chromosomal arrangement were specific of the chromosomal forms, but other could be shared between the chromosomal forms. So a full clear idea of the isolation between these 5 chromosomal forms could not be grasped based only on chromosomal data. Here it's the final picture, the different species of the complex. And if you focus on gambiae sensostricto, you can see how different regions going from the south to the northern savanna are characterized by increasing number of inversion. And you can see the arrangement that define the 5 chromosomal forms. Even in this case, of course, with the start and the development of molecular biology, we try to identify markers to identify this chromosomal form. And we published this first paper based on a mutation on the IGS region, which could identify the 3 most widespread chromosomal forms, which were forest, savanna, and mopti. And this first work was based on sample from northern savanna areas. But then when we apply the same tool to a larger geographical area, so when we move from northern savanna to all West and Central Africa, we realized that there were always 2 units that never or very rarely produce mated and produce progenies, but that the relationship between the chromosomal forms and these 2 units was not univocal. And to make a long story short, we identified we named 2 molecular forms, which were called MNS, some of which in some geographic area have specific chromosomal arrangements, but in most cases they share the same chromosomal arrangements, but at different frequencies in different ecological areas. So the bottom line of this stage in 2001 was that the Anopheles gambiae sensostricto seems to be including 2 taxonomic units, the molecular forms, which very rarely mate and are characterized by different frequency of the same chromosomal polymorphisms. And this was the pattern that we had in 2002, working on data from several Africa region. And these 2 forms were present in West and Central Africa, while in East Africa only the S form was present. So this is another representation of the chromosomal arrangement typical of the different chromosomal form. How do they fit with the 2 molecular forms? And I like only to highlight here that the 5th molecular form, Bissau, was the less clarified one. So we really didn't know at the stage whether the Bissau chromosomal form, it's chromosomal variants or one of the 2 forms or something different from both of them. Here you can see an updated distribution, respect with the previous one, not to now, of the distribution of the 2 molecular forms. And we started, thanks to this molecular tool, this PCR tool, which allowed to identify the 2 forms in all stages of the mosquito development. We start acquiring a lot of information on them. First of all, we studies, particularly from the group of Frederic Simard and Carlo Costantini in Burkina Faso and Cameroon. They show that there is a high specificity of the 2 forms for different breeding size. So the S form being more adapted to rain-dependent breeding size, while the M form is more adapted to permanent breeding size created by human activity, like in this case you can see rice cultivation. Also, we wonder what the mechanism that kept these two forms isolated in nature. Because when you put these two forms in a cage in the lab, they mate freely and they produce viable progeny. But on the same time, we didn't see many of these hybrid individuals in the field. And there's been a lot of study on the reproductive isolation, and that's been now clarified that there are no post-mating mechanism like stability, like we see, for instance, between Gambiae arabiensis and other species, but there are pre-mating mechanisms, mainly swarms, which are specific to one or the other form. So actually there is a low rate of meeting of males of one forms with females of the other form, and probably other pre-mating mechanisms that complement the swarm segregation. And basically, the general view is that we observe less, I think this is an error, less than 1%, not less than 0.1, but less than 1% hybrid individuals in the field. And here you can see very interesting work on spatial swarm segregation by the group of Tavi-Lehmann and Diabate, showing it in the single village, repeatedly, day after day, the two forms males of the two forms form swarms in different sites associated to different visual markers. And also, in addition to these ecological studies, there have been, after 2002, a lot of new studies exploiting novel, at the time, approaches to have a more clear view of the genomics of these species. And what was shown that there are quite similar, but differentiated in some specific area of the genome, particularly close to the centromeric areas. And in particular, the centromere of the chromosome X was believed to be important in the reproductive isolation. This was very nicely shown by the group of Fred Trippet by crossing different strains. So we are now, there are 10 years and more passed since the first description of the molecular the molecular forms. And in 2010, the scientific environment thought that we had enough information and evidence that these two forms are indeed separate taxa, and so they were provided as a Linnaean name, and they were raised to the species level. And the molecular form has maintained the name as Anopheles gambiae, and the molecular form was named Anopheles coluzzii, based on to celebrate the seminal studies of Professor Coluzzii. Let me stop just a second. So here you can see more than 10 years later, the first description or the formal description of the two maps, the most updated picture of adult samples collected since 2001 across West and Central Africa. These results comes from a systematic review that Carlo is doing in collaboration with us. The manuscript is in preparation and includes more than 1,300 georeference entry of adults collected directly in the field. And what is interesting, and I would like you to note on this, is that sorry, but that doesn't move anymore. Okay. 14% of these entries include individuals which show an IGS heterozygous. And I would like to remind you that IGS is the region of the genome which, based on which the two species are identified since their first description. So 14% of these records include at least one specimen showing an heterozygous genotype, which have been always considered as putative hybrids between the two species. But if you see the distribution of these putative hybrids across West and Central Africa, only 2.6% of the entries in Central Africa present hybrid genotypes, and for an overall frequency of 14%, while these increase a lot in West Africa, where the frequency of hybrid is average frequency is 0.5%, but increases very, very much in what we call the Far West region, including Senegal, the Gambia, and Guinea-Bissau, where almost 40% of the entries include hybrids genotypes, and with average frequencies of 5.7. And so the question was, this evidence accumulated over time, and the question was whether this high frequency of hybrids in this Far West region would be attributed to hybridization, higher rate of hybridization between the two species in this specific region, or to ancient polymorphisms. And here you can see there was a lot of interest on this subject, and by our group, but also other groups following the first reports of high frequency. When I mean high, we have, for instance, in Guinea-Bissau, up to 20% of hybrid genotypes. And at the end, in 2017, we published a paper in collaboration with many groups, including also Dominique, Martin, David Whitman, and Alistair, where we hypothesized that in this specific Far West region of Africa, a massive introgression led to the formation of an hybrid species. And we wonder whether this hybrid species was stable. It looked like it was stable because these hybrid genotypes were found for 20 years at very high frequency, and what kind of phenotypes could this form have, and whether it could have some relevance from the malaria transmission perspective. And now we come to more recent data that you are probably more familiar with the paper published in 2021 by the Anopheles Gambia Genome Consortium, where these are the samples that were available at the time. And you can see that the two populations from Far West Africa were not named as Gambia or coluzzii, but were left here in gray and were defined as uncertain. So that's why, because the markers that usually identify Gambia and coluzzii across Africa, you can interpret this blue, it's Gambia, and red is Arabiensis, and these are different individuals and different SNPs, species specific. They are called ancestry informative markers. So while in most of Africa, the population are strongly differentiated by these markers, in the population from Guinea-Bissau and Gambia, there is a high level of polymorphism. You can see a high level of polymorphism also in the Kenyan population here, but this is another story that we are not going to cover in this seminar. So I want you to focus on this population, which confirmed that what we saw, what we think about based on a single marker on the IGS region, so that there was something strange going in the geographic region was confirmed by much more markers across the whole genome. And here, always in the same paper, are the results of the PCR analysis based on more than 1,000 individuals, where these two populations from the Far West were clustering with Anopheles coluzzii. So this is the end, let's say, of the background. So let's see what we did starting from this information that we had available. By using RG3 data, which included more than 2,700 field specimens from 19 countries. But what we did was not to use the whole data set, but to focus on 1,190 field specimens collected in West Africa, including the Far West region. So we restricted to the analysis to the area of West Africa. And then we repeated the PCR. And you can see that we have results quite different from the previous one that I showed, because in this case, the three populations from Far West, which are the green one and the orange one, were separated in PCR from gambiae coluzzii, in particular the green one that we call GX1, gambiae complex X1, no? Gambiae complex 1, cluster separately from very far, let's say, from gambiae coluzzii, while the GX2 clusters closer to coluzzii, but separated by the other population, West African population. So, and we also ran admixture, and the results were quite consistent, because here you can see in red coluzzii, in blue gambiae, and here the three Far West population, the GX1 from the Gambia and Guinea-Bissau, and GX2, which shows a higher degree of a high degree of admixture with coluzzii. And so we try then to understand a little bit the degree of separation of this population from the other population from the rest of the range, and to speculate a little bit on their possible origin. And I should remind you that originally, not based on this data, but on previous data, the hypothesis was that this population could be the results of hybridization between gambiae coluzzii in the region. I also want to stress that these results that I'm showing are referred to data to SNPs on chromosome 3, which usually is the one that is less linked to the speciation between gambiae coluzzii, so more neutral, and most studies are always focused on these chromosomes. But we repeated the analysis on the whole genome, or on the whole chromosome X or 2, or in specific regions that of chromosome X and 2. And the results were always quite consistent. In particular, the GX1 population were always clearly separated by gambiae coluzzii. Here you can see the Hudson FST matrix showing that the three Far West population, here the first three columns, show high degree of separation from gambiae coluzzii. In particular, the level of differentiation between GX1 and coluzzii, and both coluzzii and gambiae, is as high as between the two species. While in the case of GX2, there is a very high level of differentiation with gambiae, and a much lower one with coluzzii. So at least in the case of GX1, we see a level of differentiation which is consistent with interspecific one, the one that we observe between gambiae coluzzii. And this leads us to start to show that they are really something different, strongly differentiated, and from what we knew before. And here you can see we try to analyze to understand whether this far west cluster can be considered stable populations. So we compare the folded site frequency spectra of Anopheles coluzzii and Anopheles gambiae to those of the three far west population. You can see that there is no big difference, showing that there is an excess of rare variants that can suggest a population expansion equivalent to that observed in the two main species. We did not put the graph for the Kenya population, which is the other, let's say, strange population in the data set. But if you see that population, you don't have this shape of the curve, but you have like a flat line, which shows that there is a very low number of rare variations. So apparently, the ST population are as stable as coluzzii and gambiae. And then we wonder what this history of this population, and we went straight to the main question that we always had. So these particular population are the results of hybridization between gambiae population, can be considered hybrid population, hybrid forms derived from hybridization between gambiae and coluzzii, and results of F3 statistics, always on chromosome 3 data, provide zeta values much higher than, much lower, this is a mistake, much lower, sorry, than -5, which suggests that there are no evidence of origin of this population from hybridization. So I'm not consistent with our working hypothesis that this population can be results of hybridization. And so this is like contradicting a former hypothesis that this could be the phenomenon occurring in the Far West region. To try to understand better the origin of this Far West population, we focus on GX1 from the Gambia, which look from their mixture analysis, the more pure cluster. And we use DADI, the diffusion approximation for demographic inference, to test 4 alternative hypotheses. The first hypothesis was again that this population was the results of hybridization between gambiae and coluzzii. The second one was that was a simultaneous split between the 3 groups. The third one was that there was an initial split between gambiae and coluzzii, and then that the GX1 split later from gambiae, and the fourth hypothesis was the opposite one. So first split between gambiae and coluzzii, followed by a split of the Far West population from coluzzii. And results of the analysis show that the most plausible hypothesis is most supported hypothesis is the contemporary split between these 2 groups. And of course, in this analysis, which I'm not very familiar with, but Carlo did all this analysis, you can test several hypotheses. You can introduce migration, which we didn't. So it doesn't show that this is the perfect scenario, but among those tested is for sure the most supported one and consistent with the other results that we obtained. And finally, we also carried out patterns on this statistic, the Baba test, to show whether there was an excess of ancestral of the rival alleles in the Far West population, having Anopheles christi as an outgroup. And when we tested this kind of configuration, we had an excess of ABBA against BABA, and which means that these results support the existence of gene flow between the Far West populations and Anopheles coluzzii. While when we tested the opposite scenario, we did not find any evidence of gene flow between the Far West cluster and the gambiae and Anopheles gambiae. So in other words, these results of the analysis support the gene flow between the Far West population and coluzzii, but not Anopheles gambiae. And I should here stress that when you look at the X chromosome of this Far West population, they are much closer to gambiae than coluzzii. So this is something that is relevant to always remind. So in summary, PCR and mixture separate the Far West population from Anopheles gambiae and coluzzii, and the F3 analysis did not support the hybrid form hypothesis. In particular, GX1 from the Gambia, the gene pool of GX1 from the Gambia is the most differentiated from West African coluzzii and Anopheles gambiae, and we name here as Bissau molecular form. Why Bissau molecular form? Because if you remember one of the first slides that I showed, one of the 5 chromosomal forms that Professor Coluzzii was identifying was called Bissau chromosomal form. It was exactly described in the same geographic region where we are working now. So as in the case of the other chromosomal form and the coluzzii and gambiae, it is not clear the relationship between this chromosomal form and this Bissau molecular form. But to me, that I'm working on this association between inversion data and molecular data, it really looks very much as the story of that eventually led to the description of gambiae coluzzii. So evidence from chromosomal data of non-panmetric population in some geographic region based on inversion chromosomes that are parallel by the evidence of some isolated population based on molecular and genomic data. And these are the chromosomal data based on which the existence of the chromosomal form Bissau was hypothesized. The results of that analysis suggest that Bissau molecular form maybe it should be a distinct lineage from the same population that produce Anopheles gambiae and Anopheles coluzzii. You cannot really say that it's simultaneous, but it's not too distant, the separation of the 3 lineages. We cannot really say at this stage whether it is a truly simultaneous split or whether the original divergence has been obscured later by gene flow. The GX1 population from Guinea-Bissau, seems to be the results of hybridization between the Bissau molecular form and Anopheles coluzzii population, particularly the coastal one. So what we, based on few molecular data, originally identify as an hybrid form, can still in Guinea-Bissau can still be a hybrid form. It is not anymore a hybrid form between coluzzii and gambiae, but more likely it's an hybrid form between coluzzii and the novel Bissau molecular forms. And finally, the GX2 population appeared to be a local variant of Anopheles coluzzii, and in fact now in the vector observatory is classified as Anopheles coluzzii, not anymore as an intermediate population. So now that more data became available in the vector observatory, it became clear that it's only a local variant and cannot nothing really separated. Sorry. So this is a summary of the results, but it's not the end of the presentation. I don't know how much time I still have. Antonio?
We're good. You got a few minutes.
Okay. So here you can see, so all the results that we got so far are based on 3 populations, 2 from the Gambia and 1 from Guinea-Bissau, that we have data, sequence data available. But in the meantime, many other populations have been sequenced by the Vector Observatory. This population were provided by colleagues that co-author the paper with us, and which are very important because they show, here you can see from the map in the Gambia, that the Bissau molecular form is present all across the Gambia. And we have preliminary evidence that the same also for Senegal. And this population that were collected much later, our population was collected 2005, 2006. But here you can see population collected 15 years later, and they all clustered together when we did the PCR again, including also these populations. So this seems to suggest that the Bissau molecular form is widespread and stable in Far West Africa. So why this is relevant? But this is relevant. We know from history that the presence of cryptic taxon within the gambiae complex has an epidemiological impact. The first applied consequence, for instance, can be, for instance, in interfering with control intervention targeting the main vector species. For instance, in the case of gene drive, that are very species-specific approaches, and then the presence of unknown, unrecognized cryptic taxon can, of course, be detrimental to the effect of this kind of intervention. We cannot say much more on this because we don't know anything about the biology of this new molecular form, but in fact we can say something more, because by analyzing the main genomic signature of insecticide resistance in Anopheles gambiae and Anopheles coluzzii populations, here populations available to the Vector Observatory, where the frequency of this mutation, KDR mutation, and copy number variants are summarized in the different population. Here you can see that the 3 Far West population do not show this signature, genomic signature, which are widespread in Gambia and coluzzii. And particularly the last 2 columns are referring to the Bissau molecular form. And you can see that at least at the time when these our samples were collected in 2005 and 2006, these widespread polymorphism were not present, indicating that suggesting that they may have different insecticide resistant mechanisms. So based on these few, let's say, phenotypic, genotypic, but information that lead to some relevant phenotypes, we identify some next steps that we think would be interesting to pursue in the future. First of all, understand the ecoclimatic determinants of the Bissau molecular form spatial and seasonal distribution. And already there are a lot of data in the new populations that have been provided to the Vector Observatory, which could be used to make this kind of study in Far West Africa. Second objective could be to understand the relationship of Bissau molecular form with other sympathetic species of the complex, the Anopheles gambiae complex in Far West Africa, not only coluzzii and gambiae, but also Arabiensis and Melas, which are present and sympatric in the same geographic region. Clarify the Bissau molecular form demographic history. Of course, look for genomic signature of insecticide resistance in more recently collected Bissau molecular form population, because the one that have been analyzed in our work were old, let's say, populations. And finally, characterize, and I think maybe this is least but not last, characterize the Bissau molecular form larval and adult phenotype, and the role of this form as malaria vector. To do this, however, we cannot rely only on full genomic identification of the single specimens, but we need a molecular diagnostic tool. And we provided the first step towards this goal. Here you can see the 200 SNPs which identify, which are almost fixed in Bissau molecular form and are, as opposed to gambiae and coluzzii, they are mostly in two genomic regions in chromosome 2 and chromosome 3L. And these were used as here this picture. It's the parallel of the ancestral polymorphisms that we are used to see for gambiae coluzzii. And so the 200 SNPs in the bottom line is the Bissau molecular forms. These are the region where the higher number of SNPs are mapping, and you can see that these are differentiated from how they are differentiated from most population in West Africa that have been sequenced so far. So this can be the basis to identify the most stable SNPs, identifying Bissau, and to produce easy to use molecular tools, which would allow to genotype a lot of specimens and to start understanding the larval ecology, the adult behavior, and the malaria competence of this molecular form in the field. So now it's really finished. This is our paper where all the colleagues that contributed, including also Chris and Kelly from Oxford, but also Alistair, of course, and David Whitman for Liverpool School, and Jacob Tennyson from Broad Institute, who were all instrumental for this long-standing effort that we did. It's a story that we really started a lot of time ago, and we are very pleased to see it eventually published. And so now I can thank you for your attention, and I leave space to the questions and to Carlo and Beniamino to answer the questions. Thank you very much.
Oh, thank you, Ale. That was amazing. Yeah, it's so cool to see where we've got to, but your historical and scientific scene setting is very cool. This story's been going on for so long, and how our working hypotheses have changed as we've as data has been added. Right, so we've got a question in the Q&A, and we've got some in the chat. Okay, so Shree's in the chat. Please leave your questions. Okay, so folk could leave their questions in the Q&A box or raise your hand, and then you can unmute and speak. Let me go to the... So we've got a question from Alistair. Shree, if I just click answer live, can Alistair speak? Is that how it works?
Actually, Alistair would need to be unmuted, but I can do that right now.
So Alistair, do you want to ask your question?
Hi, yeah, absolutely, sure. Well, thank you so much, Ale, for the wonderful presentation. Just like Chris said, it was a fantastic tour de force of history and current thinking. So really, really enjoyed it. And I was just curious whether there was any work on from the original work on the Bissau chromosomal form, whether anyone had looked at whether it was likely to be transmitting malaria were there any data, for example, on sporozoa infection rates within the Bissau chromosomal form, or really what was known about the bionomics and vector competence of the Bissau chromosomal form, if anything?
I should say that the Bissau chromosomal form was the most neglected, I would say, among the 5 chromosomal forms, because it was restricted to a small area where coluzzii was used to work in the late 70s, early 80s, but then he focused much more on savanna areas like Mali, Burkina Faso. So since then, I think nobody really looked. So I'm not sure that there are really data. I don't know, Benjamin, if you remember.
No data are available as you said, asking for competence. So no data. We have only data from malaria transmission in the area, but the problem is we don't know if the association of the area and the species distribution of the Bissau molecular form. But
and also, sorry if I interrupt you, Benjamin, but also it's clear that it's sympatric with other species of the complex in Far West Africa. So only knowing the transmission of malaria, the epidemiology of malaria in the region doesn't really provide any clue of whether this is a good or a bad vectors. And at the time, where few years coluzzii was working in that region, I don't think he focused on the malaria transmission aspect of the work, because he was fully focused on the taxonomics, let's say, of the chromosomal forms.
No, that's very, very interesting. Thank you so much, Ali.
Thank you, Alistair. Nice to see you. I don't see you, but still to hear you.
Yeah, and it sounds like that you're well on the way to developing an assay to be able to pull these out in larger samples. So hopefully that work isn't far away now. We'll start to dig into actually sort of things like the epidemiology and stuff soon.
Exactly. Yes, that what is needed. If you don't have an easy genotyping tool, you cannot really answer to the question.
Yeah, particularly like as you say, the sympatric. So you can't even sort of pick a region and be confident that you're looking at the Bissau molecular form or coluzzii. Cool, thank you. Are there any other questions? You could, I think you can put your hand up, or you can write in the Q&A box. We did have Ilya Dams said, great presentation. I wish to request the slides if possible. I don't know how that works, Shri. Well, I know the video will be going up somewhere, won't it? Is that?
Yeah, so the recording will be up on the Malariagen YouTube channel later today, and you'll be able to watch the recording back.
Cool. So maybe that you could use that, Ilya. I don't know. Otherwise, it might be worth reaching out to Ale if you'd be willing to share the slide. Oh, have we got another Q&A? Oh, yeah, we've got a question from Joel. Do you want to unmute Joel, or Joel unmute, and then we can you can ask your question.
Hi, thank you so much for the really great and insightful talk. I mean, you mentioned just briefly about the GCX in the coastal Kenya, but you said it's a whole different story. I was wondering to hear a little bit about that story.
I think Chris can answer this more than me, because we focus on the Far West, but I know there are other people focusing on the Far East. So Chris, maybe you can answer better than me.
There's just not the amount of evidence or data or decades of people looking at it, unfortunately. It's a different story, like as you sort of alluded to, Ale, in when we were looking at ancestry informative markers, or you were looking at allele spectrums and stuff, that it looks like it's a very odd population in the Far East. It's got very low diversity. Alistair has just put a link in the chat to a preprint by Sophia Mwingi, which looks at these populations in Kenya and Tanzania. So folk, Joel, I don't know if you've had a chance to have a look at that. But that's looking at what might be going on in that part of the world.
It certainly is a very different situation, I think.
Yeah, you see sort of quite sensible levels of, or quite normal levels of genetic diversity in the West, but in these populations in the East, they're like very low genetic diversity. So they're sort of seems
If
to be
I
quite-
can add a last comment, Chris, something that I always like to stress that I think is that after the split between gambiae and coluzzii, this is the first really strong evidence of an additional cryptic taxon within Gambiae sensostricta. And despite the fact that there have been other reports of cryptic population, but this is the first, like the Gundri form in Burkina Faso, and but this is the first time where adult populations were described. In the case of Gundri, they were always only larvae. And also the fact that this we have evidence now that this form has been there for more than 20 years, at least were reported in several in a much wider region. So I think that's really the first strong evidence of a new cryptic taxon, further complicating the picture, which is already complicated by itself.
Yeah, just call them all gambiae and have done with it. Right, I think we're going to run out of time. Does anyone have a quick question? We've got about a minute left. If not, then yeah, just to thank you again. That was such a cool talk. Thanks to you and to your team, you know, for filling us in on the whole story, right up to where we are today. Yeah, thanks very much.
Thank you to you for giving us the opportunity to talk about our favorite stories so far. So
Cool, and thanks everyone
let's hope that
for
we
coming.
can have a second episode in a few years at least.
It will be very interesting to see.
Okay.
Okay,
Thanks,
bye.
Bye-bye.
Thank you very much.
Thank you.